Right, it's 6 o'clock, so I'd like to welcome you all. Thank you for joining us for this webinar all about viral persistence and the Attomarker test. My name is Dr. Ben Sinclair. I'm a GP from the UK with 20 years experience, and I'm joined by my esteemed colleague, Professor Andrew Shaw from the Attomarker project at Exeter University. Andrew, would you like to briefly introduce yourself?
Thank you, Ben. Good evening. Yes, I am a prof at the University in Exeter, and Attomarker, for transparency, is a spin-out company from our research group that has commercialized the technology. And that means the test that we've developed in the research group is now available and can be made available in larger numbers.
Now, we want to welcome all different people who might be joining us today, whether you're a long COVID patient, a carer, maybe you're an advocate, maybe you're a clinician or a researcher like myself, or maybe you're a politician wanting to understand the national and global significance of these times. A couple of points of order. We are running for an hour, and we hope to answer lots of questions you may have. And so if you do want to ask a question, please use the chat function and direct them towards the host. Emily will be guiding us in our questioning. The format will be that we'll be introducing the test, understanding what the significance of this test is and the background to it, and then we'll be talking about some of the possible therapeutic options that come out of the Attomarker test in the context of long COVID. And to set the scene, I just want to tell you a bit about the background that I had. I was a GP for 20 years working in prison. I caught COVID in a big outbreak in 2020, came very unwell with long COVID, and fought my way back to good health. And as a result, ended up becoming an interested clinician in long COVID. We started one of the earliest online clinics for long COVID in the UK at Dr. Finlay's private practice in July 21. And since then, we've developed a multidisciplinary team of clinicians, doctors, psychologists, physio, nutritionists, naturopaths, and myself. And we use a multidisciplinary team to treat people with long COVID online and in our clinics in London, Birmingham, and Leeds. To date, we've probably treated over 1,000 people. And this conversation came about because we were looking for answers to the question of viral persistence. So the theory is that you get COVID, but you're not clearing the virus in some way, and therefore you have ongoing symptoms. And long COVID by nature is a prolonged response to the acute COVID infection. And so we were really looking for answers in that area. We were introduced to Andrew of Attomarker, and we've been using the Attomarker test at Dr. Finlay's with very interesting results to help us determine if our patients in front of us have viral persistence, and that leads us on to various therapeutic options. So we really wanted to share some of the data from these tests, which is quite an exciting innovation, which Andrew and his team have developed. And we wanted to talk about what those possible treatment options are, because this is pretty groundbreaking. We haven't really had this level of testing or understanding of a virus so far, probably in history. And because COVID's been so tested and understood in detail as the pandemic unfolded, we have a lot more data now. And I feel we may actually begin to ask and answer some of the fundamental questions about chronic fatigue, ME, and other viral, post-viral conditions through researching these subjects. So I'm really delighted to be working with Professor Shaw, and I would like him to introduce the background to the test, the story behind that, and some of the science. So Andrew, would you be happy to present some of this to us now? And we can then talk through, and any questions that come up, we can fill as we go along.
Certainly. Okay, so I'm going to share something now, so hopefully you should be able to see the presentation. And feel free to stop me at any point if I sound like a university professor giving a lecture. It's important that we should be able to explain.
Thank you. We'll try and keep it relevant to all parties. Yeah.
Yes. So this work started with Philip James when he was a PhD student in the lab. That was doing some work early in the pandemic. We spent some time even up at St Thomas's Hospital when the initial shutdown happened. But the test really came about from the idea that as a scientist, and it's important to remember I'm a scientist, not a clinician, I wanted to understand mechanisms. I wanted to understand how things work. Fundamentally, as a scientist, A plus B must go to C plus D, and you need to know what's happening. So this is the infection. This is the course of infection when you get a COVID infection. You get initially exposed to it, and assuming it comes into your body and the initial protection in your nose is not so good enough, after a few days, you'll then get an acute infection. Once you have the acute infection, all of your body's immune responses get triggered. The cell response, the antibody response, everything that helps you fight that infection, hopefully to a full and ongoing recovery. It might take a while to shake them. You can see here indicated on this picture 4 weeks or so. But then this new phenomenon has been observed of the post-acute sequelae. So after an extended period of time, symptoms aren't going, they're hanging around. And they can be in the lungs, they can be in the digestive tract, in the heart, brain, in fact, anywhere in the body. So what's causing this? Where's my mechanism? And of the mechanisms presented there, the one I like, the two I like are the viral reservoir, which means viral particles active in small quantities around the body are still there. They're not being cleared out by the immune system for some reason. And also autoimmunity. For some reason or other, you've made good quality antibodies to the virus, and that's triggered an interaction with part of the body that's normally there, like a heart muscle or a vascular muscle, and that's triggered a response, an immune response against that part of the body. So if you think about that as the cause, or those two causes, we're now getting to the point where we've got a series of symptoms, 250 symptoms are associated with this long COVID syndrome. And if you allow my virus to be anywhere around the body, you can begin to explain all of these. You don't get all of them. In fact, the top 25% here in this diagram are the mood problems and the fatigue problems and sleep disorders, but headaches, cognition, brain fog, gut problems, respiratory problems, things in your vasculature, all these other symptoms, 250 of them are cited. Now, I think my mechanism allows for all of these to be caused by the virus to be persistent around the body. So the virus, bottom right here, has got this spike protein around the edge of it. The virus is about 90 nanometers in diameter. That's quite important because it's what, 100th the size of human hair. And it's hard to find. But it docks onto parts of your body through the spike protein, and the parts that it docks onto are these ACE2 receptors, which we may have heard about now, and they're everywhere around the body. You can see in green those areas in the body where the receptors are in high density. So they are in the gut, they are in your liver, they are in your heart, they are in your lungs, small amount in your head, which means if there was a small number of virus particles there, in principle, a little micro-inflammation event could be occurring. And for me, that's a potential mechanism for explaining the symptoms associated with long COVID. So I like that as an idea. And how can I test that? Well, the spike protein, as seen in the top right, is the key, I think. So it locks onto the ACE2 receptor, but it's also floating around in the body, and you make antibodies to this. Now, that's part of your innate immune response. And the red dots in the top right there are mutations. So the virus has been changing a lot. So you may make something that sticks to the blue part, that was the original antivirus, but as it's changed, it doesn't stick anymore. So you can't just measure one antibody. That's what's available in current testing. So we invented something that might be available, able to test more variants. So imagine the blue dots here now represent the viral persistence associated with your symptoms in the brain, in the lungs, etc. If you have good quality antibodies on the left-hand side, it sticks to the virus, and the antibodies are there in purple, the virus is in red. If it sticks to the virus and hangs around for long enough, to a few hours, then the white blood cells, the phagocytes, will come along and they will clear it up. So for me, a definition of a sterilizing serum is, a sterilizing blood is a blood with enough good quality antibodies to go around the body and collect all the bits of residual virus. You could go looking for it. People have tried looking for it. That means sampling bits of the body and looking for the spike protein or looking for the viral RNA. And it has been seen, but it has been seen in people that are getting better as well. So it's not like the presence of the virus or the presence of the spike protein or the messenger RNA is indeed a good measure of whether you're getting long COVID or not. So there's my hypothesis. What could I measure in the blood that would tell me that the sterilizing process can work and that will clear the virus from the body and so improve the symptoms? Okay, so we've all been exposed to COVID at various stages. Some of us in the Wuhan wave here at the beginning in grey, that overlapped with some Delta and Alpha variants. The big wave in the middle was around Delta. And then latterly, we've been looking at Omicron and its subvariants. So the virus is a moving target. 12 variants are in our test at the moment. And what if you were exposed to Wuhan? You'd have Wuhan virus potentially persistent in your body. But also during infection, you may have made your own variant, which is intriguing. And that may be important. But what you'd like is a good immune response to all the variants. That would be the winner. That would be what a good vaccine would do. And that would then clear your body if it was in the right concentration.
Andrew, can I just ask, the graph was very interesting with a massive spike around January 2022 in the top graph with case numbers.
Yes.
Which that was when the Omicrons were, I think, in full force. Is that right?
Yeah. Also, Delta. Delta was very infectious. The transmission properties of Delta were much better than just the Wuhan variant. Okay. So Delta and then Omicron. And you can see around then there are quite a few variants out there.
And I think a lot of my patients would say that their problems did start around January 2022 with this big spike. So I think a lot of people were either infected or reinfected at that point, and there was a big spike of long COVID after that date.
Yeah, and I think it's quite important to understand what variant you've been exposed to and what may have caused your long COVID. It helps a lot with the clinical history, and it helps a lot with looking at the classification we're going to be talking about in a moment. But not everybody knows when they were sick. So it's possible they were sick beforehand, and then they got sick with another infection. Asymptomatic carriage is a problem, and it's hard to make the test specific to particular types because you don't know what is causing long COVID. Mm-hmm. So if you look at these graphs now, okay, there's a danger of this getting a little bit complex, but if you look at the four on the left-hand side, the two on the bottom represent the quantity of antibodies to all these different variants, Alpha, Delta, Wuhan, all the way up to XBB.1.5. So 11 variants here. And then we've measured the quality of those antibodies. And that means they're going to stick for long enough. They're going to hang on, and they'll make this complex, and your white blood cells go around and mop it up, and that's when clearance happens. But if I compare that with a long COVID patient, such as this one above here, you can see they first have got very low antibodies. Is that because they didn't make them to start with? They just weren't stimulated by the vaccine or by the infection. That's possible. But then look at the quality spectrum. You can see this patient here has got no good quality antibodies that we can measure. And so I would argue this patient here cannot clear the persistent virus. That will then hang around in their body, and that will give them the symptoms of long COVID. And then if you look at the one on the right-hand side, again, across the bottom, there's the perfect response. All variants got good quality antibodies. One on the right-hand side has got some good quality antibodies to Alpha, Delta, and Wuhan, the escape variant. But across the rest of the spectrum, it's quite scruffy. It's quite patchy. So if you had an Omicron infection and that started your long COVID symptoms, it's possible you wouldn't clear Omicron. It's possible that that would be the viral persistent variant. And so that would then cause the symptoms. So my test is to look at those people that have got bad quantity of total antibodies, low quantity of good quality antibodies, and the quality of those antibodies all together across the 12 variants. So that's a 36-dimensional look at people's immunity. And I think it needs to be that big because it's complex.
Andrew, can I ask just interruption there, sorry. Can you tell us about the control group and what that was made up of, just so we understand what we're testing against?
So the control, well, all of the groups are really hard. So all the people presenting to your clinic have said self-declared long COVID. Yes. And that's the best diagnosis we have at the moment. The control group that we've collected in collaboration with David Price at Cardiff University were people that we have known have had infection but are not reporting long COVID symptoms.
Right, okay, helpful. And a spread of male, female, young and old, that sort of thing?
About 50-50. Yeah, young and old is about right. They're all working in a hospital environment, so there's a slight bias there. And they're actually taking quite a lot of drugs, and normal drugs for managing their medical conditions. So there's quite a polypharmacy bias.
Okay.
But what would be a good control group? It's really hard, right? I've had COVID twice now that I know about, and everybody's had a selection of vaccines. So what does control look like now? That's almost as hard as what long
It's a
COVID
very hard question, isn't it? Someone's asking on the questions here on the chat, what numbers were in the control group? Do we know?
So we had 50 patients in the control group, and we had 150 patients in the long COVID group, 50 of which came from Cardiff, and 100 came from our private clinic patients.
Okay, thank you. Thank you. Carry on.
Okay, so what we're wanting now is a diagnostic test. And to know that you've got a diagnostic test, you've got to do a diagnostic accuracy study. And that says, I've got to recruit patients that have got long COVID. So that's tricky, right? So I'm going to take anybody that thinks they have long COVID, or I'm going to take the WHO NHS measure that says you've had some symptoms for more than 12 weeks, but which symptoms? And how do I know that you've got a long COVID symptom set that is indicative of this persistent virus? And I don't know that. So there's real problems with saying you've got long COVID. Imagine if the test were, am I pregnant or not? It's very easy to say, yes, I am pregnant. What can I now measure in my blood to say that I've got long COVID? Well, I can measure a number of things potentially, but in our test, we looked for the lack of good quality antibodies, and we took that as the measure of persistent virus. So what you do then in the test is to say, well, if I take this group of people that say they're uncontrolled and say that they're in the long COVID cohort, and I increase the level of the marker in my blood and say, how many do I get right? What's my true positive selection? So that's somebody that we think from the test is positive and they think they're positive too. Or I may measure my level in the blood and I get a false positive, which means I think they are sick, but they're not. So we started measuring the characteristics of this non-sterilizing serum, and for the 101 patients, there are 6,600 data points. And what we looked for is how well we can measure the people that think they've got long COVID. How sensitive to that are we? And we think we're sensitive to 65% of that. But lots of people may be reporting symptoms but not have long COVID. So how specific were we to long COVID? So that's 68%. And there's this magic number here called the area of the curve, which for most diagnostic tests are up at around 95% or so. But go back to the top and look at 250 symptoms. This really difficult syndrome has come out with a diagnostic accuracy of 65% sensitivity and 68% specificity. Now, I'm very happy to concede, I'm going to send this to the journal shortly. I'm very happy to concede that the referees and the journals will go through the protocol and the analysis with a fine-tooth comb. But that, for me, is a way then of working out what threshold levels for non-sterilizing antibodies are in your blood. That's indicative of the persistent virus. So if our test turns out to be reliably accurate as we have predicted, then this is what we need to see, and this diagnostic accuracy study is the beginning of the process. We also observed that some patients have very, very high antibody levels. And this is an example, right? So these patients are not in the normal response. They're not, the average response from a vaccine is around 30 milligrams per liter. This is 3 times that. So they're outliers, they're strange. They're not belonging to that distribution. And you can see this patient here is making really high concentrations of antibodies. This is almost at the antibody safe threshold for an antibody drug, right? So usually that's set at 150 milligrams per liter. So they're making, for some reason, very large numbers of antibodies, but just in these regions, just these variants, alpha, delta, and Wuhan, and they're all good quality, right? There's no problem with their antibody spectrum. So this figure we derived as an outlier at 90 milligrams per liter might be because something is triggering an autoimmune response. We measure this patient's data 97 days later on, and these levels are falling a little bit, but
still
these levels,
high.
they're still very high. and still good quality. But these other levels here are falling within a sort of half-life that you'd expect. Something is stimulating this.
Andrew, I've got a couple of patients like this, and certainly they all seem to have this, usually the first three strains that we've measured. Do you have a theory as to why they suddenly produced a very much lower response to the Omicrons that followed?
So the response in this picture here, in this spectrum here at 20 milligrams per litre is not bad for a vaccination.
It's a good response, isn't it? But I suppose what they had was a disproportionately high response to the first three variants.
So I think it's because they get a good response to the vaccine, they respond well, they make some good quality antibodies as seen here. But what you get here at the beginning is that they are continually being stimulated, and they sort of almost turn their immune system up to maximum.
Okay.
They say, right, I can make 100 milligrams per litre because this stimulus is there all the time. And it could be, I think, an autoimmune response, which means they're now beginning to attack a part of their body, a heart muscle, a vascular muscle, somewhere in their gut, maybe.
Andrew, question there about the Omicron strains. If we are being vaccinated with a later, in later Omicron territory, is it possible that then we just don't produce as much antibody to the Omicron strains anyway? I've heard that it's said that they don't produce as much of an antibody response.
Yeah, we saw that as well. Compared to the Wuhan antibody response, it's something like a third.
Okay.
That's probably because you have a recipe in your body for making the early antibody to the early variants, but the new recipe to the Omicron ones has to compete with that to get produced. There's something called original antigenic sin, which means you're good at making antibodies the first time you've seen the virus, and everything else is a bit secondary. And we've looked at that, and we've seen that, and we've seen in a cohort of teachers that if they're exposed to the virus really for an extended period of time, from the children, for example, they do start making antibodies to the Omicron variants we looked at that are comparable in intensity to the vaccine. And I think they've broken there. They've had that original antigenic sin forgiven, right? I think they're now polishing, just because it's there for a while.
Okay.
They're learning to make better antibodies.
And can I ask? I've got a couple of questions here coming in. Very, very interesting about this. This is actually the center of your work. This particular infographic. What percentage? Do you know what percentage of people had these very high levels of antibodies?
Okay, so if you look at the entire cohort, I think 65% of people have got persistent virus, and there's my confidence limits.
Yeah.
I think 13% of people have got autoimmune, and I'm still not sure about what 23% have got. So there are some big errors there, maybe up to 74%, maybe up to 21% of autoimmune, 74% persistent virus, maybe as low as 15% are unclassified. So I think the quality of the test at the moment is sitting at around that sort of classification for a cohort.
Because I think if it's a self-selecting population who say they have long COVID, we've had people come in saying, I've got long COVID, and it turns out they've got heart failure, and we correct that and they get better. So I think as we go on and define long COVID in a more accurate way and have, perhaps closer criteria, we may be able to narrow down the diagnostic certainty and the group to then test, which might help you have improved accuracy at this end of the scale.
It's hard. I think that's really hard. My feeling now is if we test somebody that has a persistent virus based on the test, can we intervene with that person and show that they respond to the intervention? That's really interesting. If it is autoimmune, then immunosuppression management of their immune system will work, and then you'll get a good positive response to that. If it's unclassified, then you're going to need to look as a clinician elsewhere. Is it heart failure? Is it some other problem? And the goal has got to be to resolve all 100%. But in the unclassified group, I don't know what can be important.
Well, I think what's interesting with the unclassified group, I see long COVID patients day in, day out, and we find that it's often more than one issue at work. There are multiple viral reactivations or new infections with, let's say, Epstein-Barr, reactivation of chickenpox virus, And all these things seem to come about, and also bacterial latent infections like Lyme disease or Bartonella, which are either activated or reactivated. And so there's multiple things going on, of which I think the viral persistence seems to be a sort of a common denominator, grinding down the immune system and allowing these other processes to operate. And my question to you is, we've had a lot of people asking questions about the how do these results relate to vaccine? So if you've been vaccinated, are these antibodies in the test produced by vaccination or by acute COVID infection? And can we tell the difference between those two strains of spike, as a wild type versus vaccine created antibodies?
So at the moment, the measurements that we're making suggest that the vaccine antibodies to all variants, if you have a good response, so just looking at those people with a good response across all the variants, the vaccine antibodies are as good as the natural response. If you look at those patients that have not made good responses, so there's a gap in their spectrum, they've got a poor response, they've also responded poorly to the vaccine. So I think the design of the vaccine, what's presented in the vaccine, has usually done a good job for both the messenger RNA vaccines and now the protein vaccines. I do think there's an advantage with the protein vaccine. So if you take the Novavax vaccine, it stays in the arm. It's a protein. It will have to be cleared by antibodies, and it'll be there for longer. And the clones will be matured better. You'll just make better antibodies. If you put the message in the arm, then your T cells get woken up as well, and they're very good at killing the sick cells. And so I think the exposure of the vaccine to the message is just shorter. So I think the protein vaccines like Novavax will give you a better chance of making better quality antibodies.
And potentially remixing your existing mix to wipe out prior subtypes that you haven't responded to. Is that right?
So that's part of, I think, the future. We can give you Novavax to fill in all the gaps and to repair your spectrum. I don't know about giving Novavax to autoimmune patients. I would want to be careful about that. That's a good clinical decision.
That might be quite challenging. And I think, a number of people do feel they have been affected negatively by vaccination. And whether that's, kicked off an autoimmune reaction that was already there, or whether that's kicked off, let's say, an immune suppressive event that allowed something else to happen, or an inflammatory event, or they just have very, very high levels of these antibodies. I think that it's interesting that actually the underlying theory that you have does seem to hold up in terms of the antibody tests, because in my patient groups, we often see deficiencies in those tests that you mentioned, where in a long COVID patient, they might produce very poor quality or very low numbers of the antibodies. And that's been a fairly consistent finding from our end at Dr. Finlay's.
I think immunosuppressed or compromised patients, I think, must be part of the group of people that are struggling to clear the virus, and indeed any virus. I think the persistent virus hypothesis, and indeed our definition here of what a sterilizing serum looks like, is going to be a problem for ME, persistent Epstein-Barr virus. As you mentioned, recurring herpes infections, I think that could be the herpes virus, recurring chickenpox. I think this is the beginning of something that we can test in a much larger post-infection environment. Could even be for post-long Lyme disease, for example. That
would be really helpful. If it's a finger prick test, because at the moment we're sending samples to Germany to test for antibodies at great expense. And the fantastic thing with Attomarker is it's quick, it's in the post, it's a finger prick test, and gives you fairly quick results. If we had that for Epstein-Barr, and if we had that for varicella, if we had that for HSV and some of the other co-infections that we're seeing, Andrew, that might really help us to differentiate quite cost effectively where people are having this immune suppression from. I think what's quite shocking to me as a clinician is that I see a lot of people, and we are testing their CD56 cells, natural killer cells, and their CD57 cells, and we're finding that they're significantly immune suppressed in one or both of those cell lines, to the point where I think as a GP previously, I wouldn't have thought of these people as immune suppressed, but actually, I think there's quite a lot of us who have been immune suppressed by acute COVID.
So I think exhaustion is an interesting idea of our immune system. If you're continually fighting things at a low level, there'll be a turnover of cells. That maybe the turnover of cells is now at a lower level. And I think part of the work you're doing with the patients is resetting those problems. If we intervene and clear the persistent virus, then I think how you reset the immune system afterwards is not easy. I think all these recurring or re-emerging viruses may become a problem. And people are also sitting on very large, complex drug catalogs now to try and suppress some of the symptoms. And how do you switch those off?
Yeah.
If I clear the virus, suddenly all those symptoms in principle could disappear.
I think what's interesting in some of my patient group, Andrew, is that I'm probably one of the few clinicians treating children. I started off treating adults, and then parents would come and say, I've got long COVID, but can you treat my daughter or my son? And I'm seeing an awful lot of mothers with daughters who are presenting both with chronic fatigue conditions. And whether there's some genetic inheritance there that predisposes to an immune suppressive event or poor response, huge correlation between hypermobility and all these sort of things that predict long COVID. And I think then the question is, whether we've got this genetic inherited risk that predisposes us to a poor response or to not clear virus. But a lot of my children aren't taking any medicines at all when they first come along. So they're very fresh, territory in terms of they should be having an appropriate immune response as a young person, and they're really not. And that's quite surprising in a young, hopefully fit, previously fit population.
Lots of things to say here. First of all, because of taking a big volume of blood from a child is hard, I would have said the whole field of pediatric immunology is way behind adults. And so that means when the immune system is evolving, we don't really know what that means. I know that when my children bring something home from school, they can shake it off in 24 hours, leaving me with it for a week. But I think I'm getting better, right? I think I've been exposed to enough passing infections now to be doing that better. The other thing is that we have reference ranges for things in our blood that vary by a factor of 5. That could be genetic.
Absolutely.
And is it possible that antibody levels in some people are genetically lower than others? I don't know, right? But I can invent hypotheses to test here, but I don't know.
No, definitely. We've interrupted on a very interesting sort of tangent, but do you want to carry on with the presentation, and I'll ask some more questions in a second.
So what can we do about this? And I think the best way to test whether we've got to a root cause is to set about some form of immunotherapy. How can we treat this? So I think the obvious thing to do would be to identify the variant that we think caused the long COVID, the onset of symptoms. So that's a clinical history point. It's going to be wrong because of asymptomatic infection and also variants you may have made for yourself during infection. Then we measure your COVID antibody test. If you have a spectrum that's consistent with immunotherapy, then we can plug the gap. If there's a hole in your spectrum, we can plug that. But if the immunotherapy available at the moment only works for the early variants, if we can repair the gap with a vaccine, that too would then begin to give you a sterilizing serum. So this is an example of somebody we've repaired with Evershot. You can see where the gap was here at the top. Antibody concentration wasn't bad. Quality was really poor. When you put the Evershot in at the first injection rather, antibody levels went up by a factor of 4, but the quality was much better. The quality is around 60%. So you can plug the gap, and the patient started getting better, started feeling better. Gut function, migraine frequency, heart rate variability. Now, because I wanted to get the virus from everywhere in the body, I needed to make sure that it got to a high enough concentration of the blood. So we, and so we worked out whether it got into their blood-brain barrier. This is an example of the Novavax response filling the gaps, repairing the gaps. So the before and after, this is a patient at the top who didn't have a bad response at all, just wanted to get vaccinated because of the increasing incidence of COVID. I've had that because I was in India recently, going to be on airplanes for a long time, so I had a Novavax jab, and some colleagues in the company also took that up. And so there was a gap in this spectrum here. Afterwards, 1 jab of Novavax, and they had, on average, a 4-fold increase across the spectrum. So there isn't an option for plugging the gap and repairing the gap. What I don't know is what recovery looks like. And that's going to be presentation of clinical symptoms, resurgence of other virus problems, hidden virus problems. But we can make a change. I can fix something that's obviously different, and I can make a measurable difference to somebody.
Andrew, can I just ask before we move on, a couple of questions coming up very relevant to this, and just for anyone listening, Evershed is a monoclonal antibody, so a synthetic antibody designed to essentially help plug the gap where you haven't produced your antibodies yourself. Is that right, Andrew?
It's a therapeutic antibody like a drug, and it was designed to help people that were immunocompromised in acute infections at the beginning of the pandemic.
And currently we have Evershed 1, which is for the early variants. So we've had various patients who have tried this medication, and we are offering Evershed in the clinic. What we find is that it can work quite well for people who have a poor response to the early variants, but sometimes in combination, the Attomarker test can help us understand whether they might benefit from a late variant monoclonal antibody, and we're hoping that Evershed 2 may come out soon. I think the manufacturers suggested it was going to be July, then it got delayed, I think, till the spring next year. So we are essentially offering Evershed from our London clinic. It's an injection, and it can be fairly well tolerated. Someone's asked about the availability of Novavax. We do also offer Novavax in the clinic. It is available privately. It's not available in the NHS yet. Evershed, the question was, would it be risky in someone who was vaccine injured? Now, the scientific sort of understanding of vaccine injury is fairly poor at the moment as a process. And so, but I have to say, I think Evershed probably wouldn't be dangerous in someone who had perhaps a reaction to a vaccine if they were trying to reduce or get a proper antibody response, because if they didn't get a proper antibody response from the vaccine, then Evershed would plug that gap, in my understanding. Is that right, Andrew?
Yeah, so I think if you've had vaccine injury, I don't really know what that means. It could be autoimmune. It could be that the message has left your arm, has gone to your heart muscles or your ovaries, that's been reported, and that you're now attacking your heart. The myocarditis problems have been reported. If you had Evershed around that, it's clearly targeting, Evershed is now targeting a different part of the spike protein. And even if you'd had vaccine injury around myocarditis, I don't think the Evershed would aggravate that. I would like to measure, well, we've measured Evershed, and Evershed 2 is indeed possibly going to be available in March next year. We're chatting to AstraZeneca about whether we can do a trial with that. I think because they're all targeted at slightly different parts of the spike protein, even with autoimmune responses, there's a good chance it won't aggravate it. That's a clinical decision, though.
Absolutely. We've got some more questions coming thick and fast now. Someone's asked where Dr. Finlay's have clinics. We have clinics in London, Birmingham, and Leeds, and we're online across the UK. And we also offer consultations around the world now. Another question. Clinically, I'm an urgent care GP and long COVID patient, and as is my daughter. I think I'm seeing both viral activation and much faster, more rapid bacterial infections. Should we be treating more aggressively when we see these infections rather than waiting to see if their body does the job? I've been doing the former because I have seen people get poorly so fast. So a clinician basically saying they're seeing people getting much more poorly, much more quickly in the post-COVID era. I would concur with that with people with long COVID or people who have had sort of post-COVID problems, because I think exactly what you're saying, Andrew, they've got an impaired immune response by nature. And so in general, their immune system is busy trying to do something else or overreacting with histamine. And so I think immune exhaustion comes to the mind, and also poor nutrition in many people is a real problem. And so we see lots of people low on vitamin D, vitamin C, and key other vitamins, which we can correct with diet and nutritional supplementation. But I do think that we probably need to be more vigilant in the post-COVID era, because I think we have a globally immune suppressed population, and a lot of us are actually immune suppressed. I reckon 1 in 10 people have some form of long COVID. And there's certainly over 2.3 million people in the UK and over 70,000 children affected with long COVID. So there's a huge problem. And I think clinicians probably should be more cautious with those patients. And it's not just like another virus. It's definitely acting a bit differently to other viruses we've seen in the immune suppression side of things. And Andrew, we've talked about Novavax. We've talked about Eversheld. Are there any more slides that you want to present to us?
I was going to say, this is the analysis of the cohort that's come from your patients in your clinic.
Thank you.
So we've tested 37 of them so far. Only 3% of them are autoimmune against this threshold. But 89% are have persistent virus potentially. 34% of those would be qualified for immunotherapy on Eversheld 1 because they're compromised in the early variants. So for want of a fairly straightforward test, we get to find out some fairly interesting information about the patients. And from now, the pictures, these are some of the patients. And you can see this patient here. Again, these 4 figures is now hopefully a familiar format. This is the good, the perfect response. This patient here has got some reasonable quality early variant antibodies, but a completely bare spectrum here past Omicron. If that coincided with the onset of their symptoms, you can't give them Eversheld for that because it won't work in Omicron, but that would indicate a vaccination. I would probably, in consultation with Novavax, give the vaccine 3 times boosted to get the antibody levels up and to maintain a good concentration and have a good persistent half-life.
So are you saying we should have 3 Novavax boosters to really boost it if we've got this sort of presentation?
Yeah, every 4 weeks or so, have for 3 boosts, and then I think you'll see this spectrum repair, and we would then measure either at the end of each vaccination or at the end of all 3 to see that it's changed, and then you get to watch the symptoms. If we've repaired it and the symptoms don't improve, at least we've eliminated potential persistence of the COVID virus. But if the symptoms do improve and we can track that, we have access to the Birmingham questionnaire that helps track those symptoms. So there's some fairly logical ways of tracking the symptoms, but I think it would become obvious. And we've seen people that have started showing improvement after 1 jab and feeling better.
Andrew, how many people have you tested with Attomarker who have had a gap repaired with Novavax, for example, under that 3 jab protocol?
Nobody's got to the end of 3 jabs yet. Somebody's got to the end of 1 jab.
Okay.
That's my problem. I don't think we have enough data to say it's all anecdotal.
Yeah.
What I would like to do is to run a study where we can look at the interventions, make sure the interventions are safe, look at what's going to be a complex cohort going in and a complex cohort coming out. What about how long does it take you to recover? If you've been ill with a long COVID for 3 years, does it take you 3 years to recover? That wouldn't be a bad rule of thumb. So we need to understand that. And we're talking to all of our clinics. You're one of our lead partner clinic, but we're talking to all the clinics around the world. And if we get adverse effects, we're reporting those. If we're getting good effects, we're also reporting those too. I just think we need to look at how these interventions can work now, whether they can be shown to be effective.
Very helpful. Someone was asking how much would it cost to get an Attomarker test from us with results with the doctor to call you. We offer that for £299. You can book that online through our website. I'll pop a link in the chat. I think the more people who get this test, the more we're going to see clearly, so the effectiveness. And Andrew, you'll have more data to go on. And anecdotally as well, obviously I'm a bit of a filter for patients around the UK and around the world. I heard of a group of doctors who took Novavax, who all had long COVID, some of whom got better and some of whom didn't. Now, they may have only taken 1 dose. I think one of them got worse. And I've got a couple of patients anecdotally who tried Novavax initially and it didn't make a difference. Now, there's small numbers, but I think what you're saying is you've got to really persist with 2 or 3 vaccinations in a regime. And I also think the background to that is some of these people may still not be having an adequate antibody response to Novavax if they are poorly immune suppressed or undernourished or end up with chronic, let's say, nutritional bowel problems or immune exhaustion. So I kind of, I think as a GP in the functional medicine sphere, what I'm trying to do is use our nutritionist, Rachel Jesse, to help boost people's nutrition and, repair their mitochondrial function to boost their actual kind of well-being before we then go forward to try a vaccine. I think you're more likely to have a better result at that point.
So I would, from the accuracy of our test, I think we'd likely to be expecting 65% of people to respond. We would recommend the vaccine to 65% of people, for example, and we would hope to see some recovery. I don't know what the other 25% are going to be doing, and I probably wouldn't vaccinate somebody who had an autoimmune response. I think that's definitely a clinical decision. It's possible it changes things, but I don't know what it does. Would I prepare the patient as best as possible for getting the vaccination? I think that's a good idea. So get some of their symptoms under control before giving the vaccination. All of those are good clinical decisions that, as a scientist, I think I can't really add much to.
I'm just popping a link into the chat for anyone who does want to book on the Attomarker test, so that you can have a look at that. We've got a question about why are we only offering Novavax, and what other vaccinations might be useful. Andrew, you seem to favour Novavax as a protein-based vaccine. Can you tell us why you think that's going to give us the best immune response?
So we have given Moderna bivalent vaccine to a long COVID patient, and they did get a good antibody response across the spectrum. So the Omicron signature derived from Moderna worked well. Moderna remains a messenger vaccine. And as I said earlier, that means you get an injection to your arm, and the cells in your arm get sick and they start making the spike protein. And that then triggers the immune response of both the antibodies and the cells. And I think your body quickly removes that because of the active T cells and good quality T cells. I took the protein because I like the idea that the protein stays in your arm for longer. It can only be removed by antibodies, and so the antibodies have to work harder to remove it. And if it does leak out of my arm, it just floats around my bloodstream and it will get chewed up as a protein like any piece of food, right? So I thought the risk of it moving around. If the message moves around, there is a risk of it arriving in the heart, as we talked about earlier, and that would then give potentially the risk of another autoimmune. Okay. So I like the fact that the protein is well targeted across, is pan-variant, as far as we can tell. I don't know about KP3 yet, but because we can't get the protein yet, but we'll test that. So it's pan-variant and it hangs around for longer. And in the patients that we've looked at, it gives a good response.
Fantastic. I've got a question here saying, how many variants are there now? I know we sort of lose track. How many variants? Obviously, we have a number of variants in the Attomarker test. Are there any plans to extend that as we get more variants?
So the number of variants is large. The number of variants of concern is still quite large. One of the ones circulating at the moment is a KP3 variant, and then you've got the flirt and the flip variants of those, they're all relatives of Omicron. And it looks like the virus is stuck in this sort of Omicron glade, as it's called. If that's true, then small, subtle variations may change the transmissibility of Omicron, but hopefully not how seriously dangerous it is. And the vaccines ought to be able to cope with that. We will extend with about a 6-week timeline. If a new variant becomes a variant of concern and we know is circulating, we'll be able to put the test on the chip, and we'll do that.
Interesting enough, for myself, I actually became better from long COVID after a reinfection with Omicron in December 2021. So I'd had 2 Pfizer jabs mandated by the government. And then I got Omicron and I just knew I was better. So I must have remixed a new mix at that point. And I sort of fully recovered after my sort of nutritional plan boosted me back to health to then have a decent response, I believe. So we're really working closely with our patients to try and boost nutrition, decrease stress, and improve the best chance of recovery. Do you want to give us another example of a story, a patient's story here?
So this is a patient who we had measured their spectrum before about 18 months ago, and they had long COVID, probably associated with an Omicron infection based on what was prevalent at the time. And you can see from their spectrum, they've got very low quality antibodies and very low quantity antibodies, but made some antibodies to Wuhan and Delta and Alpha. They then got sick with XBB.1.1.5 and made good quality antibodies to that variant. But you can see as well the quality of antibodies to BA1 and BA2 improved significantly. Their spectrum hasn't been completely repaired, but it made good quality antibodies to the persistent variant. And within a week, the patient was feeling a lot better, out of a wheelchair, no longer struggling with POTS, and indeed getting on a train and starting work again. So I think the mechanism is there for an infection to generate better antibodies and then help clear something that's persistent.
So a natural recovery. And we've got another question coming in about other therapeutics. We've talked about Evershed and MABS, synthetic antibodies. You alluded to antivirals earlier in the talk as one of the therapeutic options. We've now got Paxlovid available in the private sector. It's quite cheap, and we're giving out Paxlovid both to acute patients who've had acute COVID, which does have some evidence of helping you reduce the risk of both longer symptoms and of developing long COVID after an acute infection. But also we are seeing some positive response to people using these antivirals who have long COVID. And I think if Paxlovid is helping us get rid of persistent virus somewhere in the body, that may be advantageous. Do you have a comment on that, Andrew?
So Paxlovid works by blocking the molecules that translate the message, and it works really well. And during acute infection, it clearly helps kill those virus particles that are active. If you're a long COVID patient, I think, and get a new infection, having Paxlovid to hand would be very, very reassuring. I think that would be something I'd want to do. If I had a diagnosed autoimmune condition, so the patient I shared the study with earlier, showing the change over time, or the lack of change over time, I've been trying to get them Paxlovid for a while, so I'm really pleased to hear that you've got that, and I think they should definitely carry that around. They're going away on holiday shortly, and if to catch something on holiday,
absolutely,
and have a bit of to pack the pack, would pack the packs rather, would be good.
Pack the packs. We're certainly seeing a lot of people who fly are coming back with viral things that look very like COVID, even though they may not test positive. But we know that also some of the tests perhaps only test for earlier variants and may not be as specific for the variants we're seeing now, I think.
So the other thing is, if it's persistent in the body, it doesn't follow it's active. If it is active, then I think Paxlovid, a 15-day course or something would help well. If it turns out it's dormant and then becomes active again after your 15 days, it won't clear it. So it will clear active virus, but it wouldn't clear inactive virus. And so long COVID might just get worse again after the Pax has happened.
And a good question coming in saying exactly that you might get rebound. So I think you do need a prolonged course, longer than the 5 days for the acute infection. We're often giving 15 days of Paxlovid. If someone appears to have some benefit, we can continue that. And I think, as you say, obviously you've got to have some live and active virus rather than just a passive virus for that to be of benefit. The other thing to say, Andrew, is we are seeing great benefit from treating other viruses that may be co-infections and other bacteria that may be co-infections. So DNA viruses within the herpes group, like Epstein-Barr or chickenpox virus, respond to valacyclovir quite well. And we may not get rid of them altogether, but we certainly can reduce their activity and in that way go a long way to reducing some of the neurological side effects and some of the fatigue associated with those co-infections, and then allow the immune system more scope to deal with its COVID burden, I think, is certainly where I'm seeing that benefit. And we're finding prolonged courses of valacyclovir in the CFS community have been used over time to good effect, sometimes up to 3 months. And actually, some of my patients really do experience benefit in prolonged courses. So we've been testing and treating the Epstein-Barr and other co-infections to good effect. I'm quite excited to hear that you might be able to branch into testing for those things with a quick and easy finger prick test. It would be fantastic if you had a test, Andrew, that did a full spectrum of sort of viral tests on one finger prick. Now that would be a real winner, I think.
Yeah, we've been Epstein-Barr and the herpes virus. I think we can do exactly the same type of test, looking at variant or antigen antibody concentrations and quality, and working out whether you've cleared it or not. And as 95% of us have had EBV, apparently. Mm. The other thing about targeting that is EBV has this very oncogenic property, and it could be sitting under quite a large number of cancers. So understanding EBV is really interesting. And we've talked to the ME communities in both the UK and America, and I'd like to do that study. I think it would be easy to do. Not easy to do. I think we can do it.
It'd be accessible. Yeah.
But to have a panel that said you've got a bacterial or viral infection, we've got a new chip coming out that can distinguish between bacterial and viral infections. I don't know how low it will go in terms of a persistent near dormant infection, but if it were to recur, then we could see it. And then I like the idea of looking for the top 10 persistent viruses or the top 10 viral infections. Could we do that in a finger prick in under 10 minutes? I think possibly.
Now that'd be fantastic, because I think I'm at the moment using Attomarker really as a sort of a distinguishing guide in my management plan to say, do I think this patient has persistent COVID problem, or are they having other viral problems, or then do we look for bacterial problems? And we're also seeing a surprising amount of people testing positive for Bartonella infection, which is spread by biting insects, fleas, ticks, and carried by cats. And we are a nation of cat lovers. So we have a big Bartonella problem after a bedbug outbreak in Paris in lockdown spread throughout Europe.
Yes.
And I think also we're seeing a surprising amount of people testing positive for Lyme disease. And those people are not actually people who may have been bitten by a tick. They may have been bitten by an insect that bit the animal the tick was biting. So there's no longer just the need to be in close proximity to a tick. It may be just a biting insect. And so the other exciting thing with that is that these things are treatable. So we are successfully treating Lyme disease and reducing the other viral problems that we're seeing to unburden the immune system. And hopefully the immune system will deal with all this COVID problem as it goes along.
I think the general observation here is we're in some sort of new era of co-infection. I think
that's very true.
I think we don't just get a cold, we don't just get a bacterial infection. We need to know what we're fighting, and we need the right diagnosis.
Absolutely. And I think, I'm really hoping that as we go on, we'll be having those in the UK, not just sending, initially I was sending samples to America. We're also doing cytokine panels, but, sending these to Germany. It'd be lovely to have these tests locally sourced and at a good cost as well, because Attomarker comes in way under most of the other tests. And is quite helpful, I think, in determining, and there are therapies available to use for when you've got the test, then you know what to do. We're really excited about Evershell 2 coming out because I think a lot of our patients have this profile with a rather poor response or no quality response to the Omicrons. And I think Evershell 2 will really help them to plug the gaps in their antibody spectrum.
So I like that idea, but I still think at some point a repair of their spectrum is going to be required.
And the repair you feel would come from a vaccination or a reinfection?
Either of those, yes. I think they're the ones most likely to change your recipes for the antibodies.
Now we're winding up towards the end of our scheduled time, Andrew. So we still have some questions coming in, but I think what I would say is it's been delightful talking to you, and I want to thank all the people who are joining us from across the UK and the world. And I also want to say thank you, Andrew, to yourself and all of the team who are in the back helping all this to be possible. It gives me hope that I got better as a doctor and someone who's interested in this. And I have every hope that we will find the answers to long COVID and to CFSME in the process of these research studies, because we've never before had so much information about the post-viral era. Any final thoughts from you, Andrew? And we may still take questions after the hour is up, but I just wanted to wind up there. Any final thoughts from you?
I think you're one of the leading clinicians, having seen 900 patients. I think you've seen lots of combinations. And one of the things we're hearing from the patients is they just want to know that there's something there. They're being believed, and they're not just signing off sick from work or being forced to retire, that there's something there. And if we can measure something, and we know that it's different, one of our patients here and was just so pleased that we found something, was then in the right state of mind to cope with it and manage it, and it's working really nicely. So adding some science behind your clinical management, I think it's a good combination.
That's fantastic. Well, it's great to be working with you, Andrew, and Attomarker, and I'm looking forward to seeing our next set of results. We will have more of these chats, I'm sure, as we go on, and I think there are so many more things we could talk about. So many of the questions that we've had through are about, in a broader sense, other co-infections that we could go on and talk about. But just would like to say thank you to people joining us, and we look forward to offering you more insights into this fascinating area, and hope that you and your loved ones will be able to get treatment and recover. If you do need to contact Dr. Finlay's, please do email us. It's team@drfinlays.co.uk. I popped a link in the chat earlier about where you can get the Attomarker test through our website. And if you'd like to sign up for Evershell 1 or go on a waiting list for Evershell 2, you can connect with us. And if you need a consultation with myself or one of my clinicians, we can book that through the website as well. Andrew, any final thoughts from you in this area? What do you see the future looking like?
I'd like to see some more interventions. I think the way to test, to prove our test, is to try some interventions and look at patient outcome. If it doesn't look right at that point, then we can stop. But we have made a change. We think we can now make a difference in the quality of the analysis, and the data look impressive. We'll see. We want to get it peer reviewed, which will happen very shortly. I know government's now looking at it as well, so let's hope we can make a difference.
Fantastic. Well, on that encouraging note, I think we have actually dealt with most of the questions. Well, there's one question here that just said, how do you get tested for co-infections? What we do is we look for antibodies to the virus or the bacteria in question. So we might use antibody serology for Epstein-Barr, or let's say you're looking for Lyme disease, which is Borrelia burgdorferi bacteria. You'd be looking for a serology, so for antibodies, IgG and IgM to those. We also have another test called the Dubledore test, which uses dark field microscopy to look at the blood itself and can see inside cells at high magnification to see whether you've actually got intracellular bacteria affecting you, and indeed you can see microclots and other co-infections like mycoplasma floating around the blood, and that's backed up by a Western blot, which is another form of antibody electrophoresis. So you can see which bands pop up to which species of Borrelia, which is the Lyme bacteria, and that can guide you as to potentially where in the body the symptoms should arise. And the treatment is mostly the same. We use antibiotics and nutraceuticals, sometimes in combination, to try and get rid of those bacteria. And I think that's pretty much most of the questions that we have we've covered. So it remains to say thank you to everyone who's joined us. And Andrew, I want to thank you for your fantastic presentation. Thank you for all the questions to the audience. And please do keep an eye on our socials and look out for more of our chats as we go forwards. But I would say that's a good finish from us, and we'd like to thank you for joining us today.
Thank you.